In this video, we're going to be going over skeletal muscle fiber anatomy. Now, as shown here, this is, say, a whole muscle, like a bicep muscle. And previously, we talked about how the muscle is surrounded by connective tissue. We have the fascia here. The epimysium is a connective tissue that surrounds a whole muscle. The perimysium surrounds individual fascicles, and so all of this in here would be perimysium. And fascicles consist of anywhere from 10 to 100 muscle fibers, or myofibers. And then the connective tissue around each myofiber is called the endomysium. And that allows blood vessels and nerves and lymphatic vessels to come in. And that endomysium allows blood vessels, nerves, lymphatic vessels to enter around these muscle cells. Now, if I take this right here, this fascicle, which consists of muscle cells, and I take one of those muscle cells out right here, this is what it would look like. And then in each side of the muscle cell that's surrounded by its sarcolemma, or muscle cell membrane, you're going to find bundles of what are called the myofibrils, which we'll see consists of these thick and thin filaments. So here's one muscle cell, and inside of this are the thick and thin myofilaments. They're going to provide the contraction of the muscle. So one area that can get kind of confusing that you want to be aware of is the difference between a myofiber and a myofibril. Shown here is a single muscle cell, which is called a myofiber. And it's called that because these cells can be really, really long. And so they look like fibers, like a protein fiber. But in actuality, they are a muscle cell. So we call it a myofiber. Inside the muscle cell, you have myofibrils, and that's these, the myofibrils. And the myofibrils consist of thick and thin myofilaments arranged in what are called sarcomeres all the way down the myofiber. So looking at skeletal muscle fiber anatomy, well, as shown here, the outside membrane of a muscle cell is called the sarcolemma. Now you're going to see sarco a lot when you're talking about muscle. Sarco means flesh. Like, for example, the Egyptians put their bodies in a sarcophagus. Sarco means flesh. A lot of sarco terms when dealing with muscle. Sarcolemma is a plasma membrane of a muscle cell. So shown here is the sarcolemma, and it goes all the way down the muscle cell. The sarcoplasm is the cytoplasm that's inside a muscle cell. So there's lots of mitochondria, there's glycogen granules. It's going to contain the myofibrils, the thick and thin myofilaments that make up those myofibrils. And then along this sarcolemma, you're going to see these indentations in through here, or extensions of the sarcolemma. And that sarcolemma continues on. So when we talk about those voltage-gated sodium-potassium channels that are going along the sarcolemma, these same channels come down through the T-tubule, because the T-tubule consists of, well, sarcolemma. It's just a particular part of the sarcolemma as it comes down, which is the T-tubule, still contains those voltage-gated sodium-potassium channels. And what we're going to see is that when action potentials come down the sarcolemma, they continue down these T-tubules to carry the action potential deep inside the muscle cell, which is going to stimulate this structure right here called the sarcoplasmic reticulum. It's a modified version of the endoplasmic reticulum. Only here, it's modified to store lots of calcium. So when we look at the sarcoplasmic reticulum, it's the endoplasmic reticulum that surrounds the myofibrils, those thick and thin myofilaments. So all of this shown here in blue, as well as over here, is the sarcoplasmic reticulum. And it functions mainly as a calcium storage depot for muscle cells. Well, there's certain parts, and we're going to see that calcium is going to be released in order to stimulate a contraction. Well, when you look at the sarcoplasmic reticulum, there are some portions of the sarcoplasmic reticulum that have expanded out. In fact, if you look here, here's your T-tubule, which is also shown here. The T-tubules come deep inside the muscle cell. Oh, incidentally, the T stands for transverse because they're coming through the muscle cell in a transverse direction. Well, as that T-tubule comes through, on each side of the T-tubule is a portion of the sarcoplasmic reticulum. Only this is an expanded out portion of the sarcoplasmic reticulum, which we call the terminal cisternae. So here's a terminal cisternae here, here's a terminal cisternae here, and with the T-tubule running down the center of this, it makes up what's called a three-bodied triad, as shown here. Now what's amazing is that when you look at a skeletal muscle fiber like this, every single one of these sarcomeres that go up and down the muscle fiber, the muscle cell, is going to have these T-tubules along each side of the sarcomere. So it's going to be a lot of T-tubules, and there's going to be a lot of sarcoplasmic reticulum in order to deliver calcium to that muscle for a contraction. So now we're going to go into more detail about those thick and thin myofilaments that make up the myofibril, as shown right here. So the myofibrils consist of thick and thin myofilaments, and we're going to see that these thick and thin myofilaments are going to slide over each other during a muscle contraction. So what you're seeing right here, beige color, is a thick filament, and it's going to have these myosin heads that come across. These are myosin heads here, and they're going to bind onto the thin filament during a contraction, and they're going to pull the thin filaments over the top of the thick filaments, and what's called a sliding filament model of contraction. So now we're going to go into more detail of what a thick and thin filament is to make up these structures here in the muscle cell, which we call sarcomeres. And basically a sarcomere just goes from Z disc to Z disc. So when you look at the myofibrils, as I said, they're these rod-like extensions that extend the length of a muscle cell. They're composed of cylindrical bundles of thick and thin myofilaments. So when we're looking at this muscle cell right here, we're going to see that there are thick filaments and there are thin filaments, and they're arranged in repeating units called a sarcomere. And we'll see that the sarcomere consists of what goes between the Z disc to the next Z disc, and you have your thin and your thick filaments. That makes up a sarcomere. So all the way down a muscle cell, we have these repeating units called sarcomeres. This is one myofiber, which consists of myofibrils in here that make up the thick and thin myofilaments, or the repeating sarcomeres. We're also going to see that this is what contributes to the striations of striated muscle, these stripes that go down both cardiac muscle and skeletal muscle. So the thick myofilaments. Well, the thick myofilaments are here, and they're going to have these globular heads called the myosin heads. This is a myosin protein that makes up the thick filament. They're also referred to as cross bridges. So when you look at the thick myofilament, it consists of about 300 of these myosin protein molecules. And each myosin protein molecule, as shown here, is made up of, well, what looks like two golf clubs, with their two heads of the golf clubs here, and the shaft of them are twisted around each other. That's one myosin protein. You put about 300 of these myosin proteins together, and you get this structure here, which is called the thick filament. So if I come back up here, this is the thick filament that have those myosin heads sticking up that are going to bind on and are going to move the thin filament over the thick filament. Well, each myosin head contains a ATP binding site. So on this myosin head, you'll find a binding site for ATP, and we're going to see that ATP is going to energize this head to cock it back, to get it ready to bind to the thin filament, in which it binds and it power strokes to move the thin filament over the thick filament. It also has on the myosin head an actin binding site. So the thin filament here, we're going to see that they consist of these protein ball-like structures called actin, and they're going to bind to the actin active site, or an attachment site, bind, power stroke, and move the thin filament over the thick filament. Well, once the ATP is used to energize the head, it's converted to ADP and phosphate. Little I there just means inorganic phosphate. That inorganic phosphate and ADP will come off, which then allows ATP to bind again. And we're going to see that ATP is needed to release the head, cock it back, which provides the energy to the head to then bind and power stroke. More details on this will be covered later when we cover muscle contraction. When we look at the thin filament is shown here, purple, is going to have binding sites for the thick filament. Well, the thin filament has a little bit more detail than just what's shown here. It looks like this right here. There are other proteins involved. So when you look at the thin filament, or the thin myofilament, it's composed of 3 different types of protein. We have actin, which is a main component. That's the purple balls you see here. We have troponin, which is in red, and we have tropomyosin, which is in this lighter blue color. Now the actin myofilament, referred to as F-actin. When you're looking at it, the whole thing together, it's F-actin. And it consists of, it's a polymer that consists of about 300 to 400 of these globular subunits that we call G-actin. And they're arranged in a double row of proteins twisted to form a helix. So all of this is going to be your actin filament. Well, on each actin subunit, you're going to find a binding site for the myosin head. And under resting conditions, the myosin binding is prevented by what's called the troponin-tropomyosin complex. So this right here is troponin, and then the belt-like structure is called tropomyosin, and these act to cover over the active sites until it's time for the myosin head to bind. So when you look at tropomyosin, this is the light blue strand. It's the primary cover of these attachment sites where the myosin head is going to bind. They're long strands of protein molecules that are located in the grooves here of the actin helix, and it covers the myosin binding sites and prevents actin-myosin interaction. Well, attached to this tropomyosin are troponin molecules. Troponin binds to tropomyosin, forming what's called a troponin-tropomyosin complex. Now, troponin is a complex of 3 proteins. We have troponin I, and basically that's this right here, also shown on this diagram here. Troponin I stands for inhibitory, and this is a portion of the troponin that will cover over an attachment site or active site for the myosin head. Now, it doesn't cover up as much as, say, the tropomyosin, but it does cover a little bit, and so this portion of the troponin molecule we just refer to as troponin I. Troponin T is this right here, and that's a part of the troponin that actually binds to tropomyosin. So shown here is troponin T binding in with the tropomyosin. And that troponin T, the T is for tropomyosin. Troponin C, C stands for calcium. This is a part of the troponin molecule shown here and shown here where calcium is going to bind. And we're going to see that when calcium binds to troponin C, it's going to cause a conformational change in shape change in troponin. And that's going to pull this troponin, along with the tropomyosin that it's attached to, it's going to move it out of the way of the attachment site for the myosin head, so a contraction can take place. So you look at the myosin heads as shown here. We have our ATP binding site that's going to cock the head back. And these myosin heads are going to bind to the binding site or attachment site or active site of these thin filaments. All right, so next up we're going to talk about the structures of the sarcomere and the striations that you see in a muscle.